tremendous expertise and knowledge in all these areas, and we welcome him here, and we look forward to the talk and the discussion.
Okay, thank you. So I'm going to give what in the end will look like a very superficial overview of a lot of topics, but I want to try, particularly for perhaps the mathematicians, to indicate what some of the fundamental problems are in terms of the biological side. Now, you may not understand much of the terminology that I'm going to throw out. You've heard some of it in Anatita's talk. That's a constant problem when you try to cross disciplines, but feel free to ask questions either during the lecture or afterwards. So, what happened? Oh, why is that not moving?
Too much adhesion.
That's bizarre. Whole thing is frozen.
Maybe log off and log back on again?
Oh yeah, but it's frozen.
Like push the button down, power button off, and then log back on. It happened to my student in Atlanta.
Well, this has not been a good trip. When I was in Edinburgh, the whole system crashed and I had to start over completely. But we'll see. Anyway, let me talk while this thing is getting itself in shape. What I'm going to start with are some of the basic principles that underlie cell motility. And then I'm going to talk about a model system that encapsulates basically all of the processes that we have to understand in order to understand cell motility. And then I'll give you preliminary results on a number of different aspects that are involved. And I'm not good at talking and typing.
Yes.
Okay, Professor Winter and Professor Hoffman.
Okay, since we're going to have to wait a minute to do that, let me here draw a cartoon. Where are the
Thank you.
erasers? Here. Okay. Cartoon of a cell. Okay? Anatita called this omega I of t. Okay? Some of the essential things that we have to understand is that inside the cell membrane, this is the lipid bilayer, there is something called the cortex. That cortex is comprised of actin filaments, cross-linking molecules, myosin, which is a motor protein, and a variety of other things. Okay? That's a static view, but what I'm going to try to show you are some examples of how this sort of structure has to be remodeled when a cell moves. I mean, we're talking about motility, and by motility we don't mean a cell frozen in place, right? We want it to move. So some of these things are going to appear for the later discussion, if we get this thing to fly. How many of you know what actin means? Anatita used that word. So, yeah, maybe a half of you, okay? So there again is the issue. From the mathematical standpoint, to do a good model, you have to learn a lot about the biology. From the biology standpoint, to understand the models, critique them, suggest improvements, and so forth, you have to understand a bit of the mathematics. Talks, cell move, Cambridge. seems to be inordinately slow. I don't know what the projector is doing to it. There. Okay, let's hope. Keep your fingers crossed. Okay. Why understanding cell motility is important in the first place. I mean, why do we care? Okay? So it's essential in a variety of different processes, starting from development of you from a single cell to the organism that you now are, to the immune system, which helps you preserve yourself as you are. In wound healing, cells move into the wounded area. You may not be aware of this, but you put a cut in your hand, and if you were able to track what was going on, you would see cells moving into that cut to fill in the gap. Angiogenesis, this is the process of forming new blood vessels. It occurs during early development. Obviously, you start off as a single cell, and eventually you end up as this body with a vasculature that pumps blood throughout it, containing oxygen, etc. But that's but this angiogenesis is particularly important in cancer, and the cell motility is frequently what makes cancer as dangerous to an individual as it is. Metastasis means that if you have a tumor, a small tissue-like structure of cancerous cells, metastasis involves some of them breaking off, moving through the surrounding tissue to a nearby blood vessel, and then populating some other organism. At that point, it becomes very serious, because if the tumor is highly confined, you can use very strong treatment methods for it. If it becomes dispersed throughout the body, you cannot. Okay? Then at the organism level, there are many other reasons. Okay? There are lots of different modes of cell motility. Bacteria, E. coli, which populates your gut, for example, they swim either individually or as swarms. There are lots of examples of crawlers, and I think most of what we hear about in the next couple of weeks will be related to crawling cells. I'm going to give you an example in the form of this movie of a crawling cell, I hope. There. Always happens at first. And I want you to think, as you watch this cell crawling, what are the processes involved? What sort of descriptions are involved? What sort of mathematical models they might lead to, etc., etc. So here it is. It's a very famous movie when it appears. There. Okay, this is a neutrophil chasing a bacterium. How many have seen it? Probably many of you. Okay. Now, focus on that guy and ask yourself, what is he doing? Let's make it a he. What is he doing as he's moving? Look at how the cell is deforming itself. It's attached to a substrate, so it's exerting force on the substrate, right? It has to decide how to move. Why? Because it's going after this bacterium, the little spot, all right? So the bacterium is emitting some kind of a signal, not purposely, but the neutrophil has evolved to detect those kind of signals. So it has to detect that signal. It has to transduce that signal into some kind of an internal cell signal. Then it has to decide, in quotes, how to remodel its body to move in the appropriate directions. And those are some of the key problems that we face when we try to describe cell motility. But it turns out the cells are also very plastic in the sense that the same cell type can use different modes of cell motility, or different modes of moving, depending on the environment that it finds itself in. Okay? Again, we're probably going to be talking most about cells that are crawling along a substrate or through the ECM, the extracellular matrix, etc., etc. But recently it's been discovered that there are cells that can move simply by effectively pumping water through themselves. Okay? And how that works is if you control channels at one end of the, suppose the cell wants to move in this direction, if you control channels that let water in at one end and pump the water out at the other end, you can effect net motion, okay? And that's an area that's turning out to be quite important in cancer, it turns out, because this is how many cancer cells move when they're in very tightly constrained environments, okay? Okay, so what are all the basic processes? I'm going to talk primarily about directed movement. That is where cells, yes?
Could you maximize the screen again?
Oh, you really want me to try this? We're going to talk about directed cell movement. Okay, there is also random movement, but that's of less interest to me at least. So there's a signal of some sort. There's a mechanism for detecting that signal. Then there's some signal transduction steps, some internal response, in some cases that I'll talk about later, signal propagation, then movement and generation of the energy required, etc. So there's a complex interaction between the biochemical networks that are involved in signal transduction, control of the cytoskeleton, force generation, and energy supply needed for all these processes. Okay? So here's what we'll try to do, but I guess I certainly won't make it anymore. I'll talk about a model system that Anatita mentioned briefly, but which encapsulates all of the processes. It's been extremely widely studied, so we know a huge amount about it, and it does almost everything that a eukaryotic, that a mammalian cell does. Okay? Now we'll talk about signal transduction and direction sensing, a bit about actin dynamics, stochastic modeling, etc., etc. But I probably won't get through all of this. Okay, so dicty. Well, here first. Here's the life cycle. When they're in a good environment, they eat bacteria, they grow, divide, etc. When the local supply of bacteria is depleted, at various points throughout the field that they're residing in, certain cells start periodically emitting a signal. It's cyclic AMP. That signal then moves outward by diffusion. It reaches neighboring cells. Neighboring cells move toward that, but they also relay the signal. And in this process, they can organize a large field. So you have, in the end, a concentration of cells here, which then goes through a variety of other stages, which we won't talk about. Well, I'll show you some of these in a movie until, when conditions are right, the cycle starts over again. Okay? So, this is a beautiful movie, and some of our German colleagues could give the translation online, but I'll give just a few comments. Okay, so here are individual amoeba. Okay, you notice they're moving around. They're on a surface. All right. Somewhat later, in one of these later stages where they've begun to aggregate, you can see them making cell-cell contacts. And at a somewhat later stage still, they begin moving collectively. Okay? So you've gone from individual motion to collective motion. But there are cells which are still attracted to this. Still later, we're in this phase that looks like a sort of cigar. It's called a slug. That slug can migrate, forms these streams. Here's the center I pointed out earlier, and you see it rotating, so there's a nice problem of what is it that causes these. They're all responding to the same signal, cyclic AMP. So there's a wave of cyclic AMP that is circulating in space. They're responding to it. Okay? And you can see various concentrations. You see here, they've reached that stage where they begin to move as a slug, and somewhat later you'll see, well, here you see it. All right? So this might be of the order of 10 to the 5th cells that are moving collectively. All right, so a question is, who's in control? How do they organize themselves so as to move collectively in this way? This is a cross section of it. It's not terribly great, but notice that this collection can raise the front end in the way you see there, and eventually it's going to erect what is called this fruiting body, which is the, let's say this, here it goes. This is the sort of erection of the fruiting body which constructs the safe place for the cells at the top. There's an interesting evolutionary question here, because the cells at the top are the ones that propagate their genes to the next generation. The ones that are in the stalk here are dead, okay? So you might imagine that there could be cell types which cheat, i.e. they produce more cells up here, which are called the pre-stalk cells, pre-spore cells, than the stalk cells. And indeed, people have found those kind of mutants which can cheat, okay? So, much like people, right? Or we're much like them, if you will. Okay, now, as I said, we're going to probably focus much on crawling cells, cells that are attaching themselves to a substrate, exerting force on it, and pulling themselves in that way. But people discovered that there are other modes that they can use depending on the medium that they're in. And here is an example. This is, again, the same dictyostelium cells, the same type of cells, put into an oil which has a gradient of density so that the cells can find a level of neutral buoyancy. Then you put a pipette into that at that level, and you ask, what do the cells do when you release cyclic AMP? And what you hopefully saw while I was talking is that the cells are moving toward that source, but they're not attaching to anything. They're in a fluid, so they can't pull themselves forward by attaching to a substrate. So what do? Well, what do you do when you're in water? You swim, right? And Dicty has discovered the same thing. And in fact, I'll just go directly, because there's a movie on the next one. And what you'll see in the next movie are the enormous shape changes that it undergoes. Now, Natita showed one example of it in this process of swimming. Watch these. Okay? Those are the kind of questions that we would like to address with mathematical models.
So what is the medium really?
This is still just that fluid. So these guys are swimming by shape changes. Okay? Now, you can swim by shape changes. I don't know. Do you swim? You swim by shape changes by doing this, right? But as we'll see later on in the talk, the regime in which these guys exist is very, very different than the regime in which Anatita swims. And so the fluid mechanics is very, very different. Okay, so in fact, Dicty has several modes that it can move by, and Peter von Hostert classifies them as gliding, walking, and swimming. In these latter two, the way it moves is, think of the basic body as kind of an ellipsoid, then it exerts small protrusions at the front and then propagates them down the length of the body. So in this context, what the cell is doing is deforming itself a bit. Let's call this the front, putting out a bit of a protrusion and then propagating that to the back. And we'll show you later that that indeed can lead to cell motion. Okay? So, three big problems. The transduction problem: how does the cell detect the extracellular signal? How are they transduced into the changes of the mechanical, or the structure of the system and the mechanical forces that control the shape changes? What I call the exterior problem. Forget about the internal changes in structure and so forth, and ask yourself, what sort of shape changes can actually lead to motion. Okay, it's a non-trivial question, but we'll see what the answer might be. And then the interior problem, basically how do you couple the first two? How do you generate the shape changes that give rise to motion? And by the way, for those of you that are interested, I can post this talk somewhere, so if you want references and so forth, you can get them. Okay, so let's think in terms of a modular approach to motility, where we separate the signal detection and transduction from the mechanics involved in changing the cytoskeletal structure. Okay, first the signal transduction. And this is an interesting area in which to work because there's an enormous amount that's understood about the biochemistry, but putting the pieces together has not yet been done. The analogy I use is the following. If I take my watch off and I want to understand how it works, I could take the back off, I could take the springs and the screws and the little gears, etc., put them on the table. Would I understand how the front works? Of course not, right? So it's the integration of all the biochemistry, etc., that is currently understood, which will lead us to an understanding of this particular problem, okay? And the details are not important here, except to understand there's a linkage between the extracellular signal, which is cyclic AMP, and through a detailed network, the F-actin, which is filamentous actin, I'll elaborate on that in a moment, which is building the structure which has to be remodeled as the cell moves, okay? So the first issue is, how does a cell orient itself? So ask yourself the following question. If you're in Cambridge on a Sunday morning and you want to find a bakery, how do you proceed? You don't have Google. You don't have any of those things. I beat you. Maybe you're near a bakery and you get a whiff of it. What do? Well, you sort of you internalize the signal where you are, and you take a step, right? And you decide, is the signal better here or worse? If not, you maybe back up. This is exactly what bacteria do, because bacteria are too small to measure signal differences over their body size, okay? But these cells are big enough. So somehow they understand, in quotes, the difference in the signal between front and back. The internal biochemical network can generate a difference in the signal at the front and the back, and this is what gives them the orientation, okay? Now, with Dicty, there's a very interesting experiment which kills a lot of the existing models, and I won't go through the details of this, but the experiment is the following. If you give it a signal, cyclic AMP, around or at the front, say, with a pipette, then the initial response is actually uniform around the cell, which sounds a bit counterintuitive, because how can it orient itself if the response is uniform? But then there's a secondary response which produces the symmetry breaking, okay? So the initial response, and this is the experimental result, this is the initial response front and back, dark is back, front and light is back, is the same, and then secondarily you get the symmetry breaking that determines the orientation of the cell. And we have a network recently that does that. I guess I'm so far behind I won't go through the details of it, but I'll tell you that it works pretty well, in the sense that if you give it a signal, you initially get a very similar response front and back, but then they diverge, okay? So the signal transduction issue we're starting to make progress on, and it involves a fairly detailed model of that network that I showed you, okay? Now, let's go on to the next step, the actin dynamics and what's involved in the cytoskeletal changes. So here's the signal, extracellular stimulus, that the cells are detecting. And that, again, induces a complicated sequence of events, the net result of which is, under appropriate condition, you begin to build a network of these filaments which can push against the membrane. Okay? So just think of this as the network, okay? And it grows, and in so doing, it can push against the network. And I think that Alex, will you talk more about that?
Not really, but
Okay, well, you can anyway. Alex is the expert, so if you really want to know about building actin networks, talk to him, okay?
are you using networks in two different ways?
I'm using networks here in the sense of this polymeric network. So,
Physical network.
pardon?
Physical network.
It's a physical network, not in a sort of graph theoretical sort of sense of network. Okay, it's this polymeric network, and I'll come to a little bit of the details of this in just a moment. In fact, I think the very next one, yes. Okay, the basics on actin dynamics. So actin exists as a monomer, and it can polymerize. What you saw in the preceding slide, where a lot of these filaments, the long polymers, link together in various ways to form this network. Okay, details. Well, all right. It's very difficult to start a network if you simply put actin into solution without anything else. And so that's the reason why on the surface of a cell you have all these additional molecules, which I didn't go into the details of, which initiate the formation of these filaments, okay? And it's an open system in a thermodynamic sense because it uses energy throughout the process to generate these filaments, etc. Okay? And there's many cross-linkers. Okay? So this is a huge problem by itself. But hopefully some of you are thinking, how can we simplify this? Well, Anatita went to what I would say is perhaps one extreme. One variable describes the whole process, but there are many more details that have to be incorporated, and some of the people here know much more about that than I do. So what are some of the basic questions? There are a large number of them. Basically, well, let's go back one step. These polymers are interesting in the sense that they do what is called treadmilling. That is, I have two ends. The monomers themselves are oriented. They have a plus and a minus end, all right? So when I build a filament, I get an oriented object. It has a pointed end and a barbed end. The pointed end depolymerizes faster than the barbed end, so you end up adding monomers at one end and releasing them at the other end under normal cell levels of G-actin, and that is what is called treadmilling. And that's what basically controls the extent of the network within that cortical region. Okay? You see, if you were to build the filaments and never destroy them, you would end up with a whole cell mass filled with this polymeric network. All right? So you have to have some mechanism by which it deconstructs, and the treadmilling is the mechanism for that. Okay, so here's where some work from Till comes into play. So you would like to understand how these processes of building and reconstructing and deconstructing networks work in cells. And so what Günter Gerisch and Till and others in that group did is to treat cells with a chemical which completely depolymerizes the network, okay? So you knock down the network completely, and then you watch it grow back, all right? And what happens when you watch it grow back is very interesting. Here we're looking at a cell from below via what is called turf microscopy. That gives you just a couple hundred nanometers at the bottom of the cell. And what you see are spots which represent actin network which is being built at that point, okay? And it sometimes evolves into waves, and those waves propagate throughout. Initially here you see a lot of spots emerging, and then later on you will see full-blown waves that emerge. And a question is, how would one describe this? First, here is a more detailed picture of the front of a wave. This is actin at the front, which is high. The wave is going in this way. Behind it, I think is pitry green in this picture? Yeah. One of the other chemicals in the signal transduction network behind the wave. Okay, so the network that's involved is very complicated, but I'm going to show you a cartoon description, okay? And again, I want to emphasize that you can learn a lot from cartoon descriptions, even though they don't contain all of the essential biology, if some of the processes are represented sufficiently. And the simplified model is going to be this, okay? We have what are called nucleation promoting factors. These molecules are known. They sit on the membrane, scar, wave, indicti, etc., and other molecules in other systems. They have to be activated, all right? They go into an active state, and that active state then can generate new free barbed ends at the network. So there's a positive feedback loop here, and once they're activated, they go into an inactive state and then return after some time. To how many of you does this suggest immediately a mathematical model? What? Fitsunaguma? Okay. Whatever your model was, Fitsunagumo is one example of what is called an excitable system. That is, it's a system which has a stable rest point in its dynamics, and if you perturb it sufficiently, it gives you a big response, but then it relaxes to back to the steady state. And if you do this in a spatial context, if you have a Fitsunagumo medium in space and you initiate it at one point, you will see a wave propagating out. Well, that's very much what we saw with the actin spots, okay? So let's see if a model can tell us anything about that. And here it is. It's very complicated, and since I'm almost out of time already, and what, halfway through?
You have another 15 minutes.
Oh, I thought I was done at 11:30.
11:40. You have an hour. I have 45 minutes off. I should have warned you, but
because I'm a slow talker.
From 5 minutes to 15. How are you doing that?
well, we need to. Yeah.
Okay, so it's very complicated. But what are the things that we have to involve? Okay, we have to have some mechanism for generating filaments at the surface. Okay, those filaments then grow. All right? Somehow, this growth of filaments has to be able to propagate. So we have species which are sitting up here in the cytosol. That's going to be G-actin, the filaments that have grown, various other proteins that are involved in controlling the network. Remember, I told you there are lots of molecules that are linking the network, deconstructing, et cetera, et cetera. And at the surface, we have these we have sites at which what we call the backbone filaments can be made. They could arise, for example, at a molecule called formin, which is known to initiate filaments. And then the network evolves, okay? The details are pretty long, and as you'll see in the model, it's pretty complex. Now, when you think back to the movie, what you saw looked very much, I would say, like a random phenomena. That is, there were spots which are emerging at random points in space. Some of them then grew up to be full-fledged waves, and then others simply died away. I'm going to first write down a continuum model of this, and then ask the question, how do we go from this to a stochastic model for the process we're interested in? In any case, what we have in the cytosol are these four components. They diffuse, they react, etc. You saw these kind of equations in Anatita's talk. Let me ask just one of the biologists. Can you parse this equation in the sense that you know what each of the terms mean? How many would be able to do that? Okay, here's what I will say, because a developmental biologist and I a few years ago ran a course exactly with this kind of audience. There are many, many technicians that can solve this equation, okay? But there aren't many people that can put it together, okay? So what you as a biologist should try to do is understand what processes are reflected in equations like this, and are the essential processes that you know exist there? If not there, why not? Should they be included, et cetera, et cetera. So I think your job should be to learn how to interpret at a certain level what these equations mean, and then, as I say, there are lots of technicians. I mean, it's very easy to solve some of these equations numerically nowadays, okay? I had one student who, when she came in, was petrified at mathematics. At the end of the course, she could tell you everything in this equation, okay? She understood what processes were leading to what kind of terms, and I think that's what's needed, and hopefully some of that will come out of this meeting. You'll see lots and lots of models, but I would hope that some of the experimentalists would pull us up and say, hey, that's rubbish. You know, you've forgotten such and such a process which would lead to another term in your equation. Okay? If we do that at the end of the 6 months, then we've been very successful. Okay, so it's a huge model. Okay? You've got species diffusing on the surface. Anatita had an example of that. You've got species diffusing and reacting within the substrate, et cetera, et cetera. And I said this is a stochastic process, but all these equations are deterministic. So how do we proceed? Can we say anything intelligent about this? Well, what we do is the following. We first do the standard thing that you do with a partial differential equation. You discretize in space. All right? Now you have a large system of ordinary differential equations, and now you simulate those stochastically. That's a huge problem, and it's led to some very interesting mathematical questions. First of all, how do you choose your discretization so as to accurately describe the stochastic process that's occurring? Okay? If you make it too big, you've lost the essence of the stochastic process. If you make it too small, you have no molecules in your domain. Okay? So there's a question arising from that. And then there's a question of what are efficient computational algorithms to do this, because this is a very large problem, okay? And that's ongoing research. We have a paper coming out soon in which some new methods are developed, but it's certainly not a solved question. Okay?
One question.
Yes?
In your case, stochasticity comes just from number fluctuations within the simulation box.
Yeah, yeah. So we're going to start, just think of a box. You've got a small number of molecules within each box, so the numbers are fluctuating. The diffusion part of it enters in the fact that I take my domain and I discretize it into a number of little boxes, and then diffusion is treated as a jump process between boxes, okay? Basically as a random walk on a lattice would be.
What's happened to the filaments?
Pardon?
What's happened to the filaments?
Oh, they're going to grow. So one of our...
I guess they're three-dimensional objects.
Yeah. So this is a simulation.
But they're not in your equations.
Sure they are. If you work through it all, they are.
Concentrations.
Because what we're doing is we're tracking the length of different objects, okay? So the evolution of the length of objects with certain numbers of monomers are in the model, okay? Okay, so what does it predict? Well, let's see. Here, we start with a small stimulus in the corner, and we let it run.
What's the unit of the colors?
So this just reflects the height in monomer numbers, and the reason we cut it off here is because the turf images really only capture that part very close to the membrane, okay? But anyway, you can see, first of all, that it's a very jagged wave, whereas if this were a deterministic equation, you would get a very smooth front propagating outwards, okay? Secondly, you see that it really does look like it is an excitable system in that we stimulated a corner and you see something propagating out, but behind it, the medium has relaxed back to whatever its initial state was, okay? To further test this idea as to whether it really is an excitable medium, oh, well, here's, in response to your question, here is the height of the monomers. I'll skip that. To test whether or not it really is an excitable medium, let's start a wave at each of the corners and ask what happens when they meet, and pay careful attention to what happens behind the wave in some places in space. Okay? So they annihilate, as you expect. That's characteristic of an excitable system. When the waves meet, they annihilate. Okay? They don't pass through each other as they would, for example, with the Schrödinger equation. But look what happened. In the wake of the wave, there is still medium which can spontaneously activate and initiate a new wave. And that's exactly what you saw in the experimental pictures that I showed you earlier. Okay, another one started over here. That's fundamentally a result of the stochastic nature of the problem. You would not see that in a deterministic equation. So the point here is that depending on the phenomena that you're trying to describe, you have to use the appropriate level of description and type of model to describe the phenomena. Okay? Had I simply integrated the PDEs as a deterministic system, I would never have captured this. But look at what happens. And if you remember, that looks quite a bit like what the experimental results
Do we
show.
understand x and y to be in the tangential plane to the cortex?
Pardon?
Do we understand the xy plane to be in the tangential plane for the cortex?
That's the, yeah, that's the bottom surface, but the color indicates the height at that point. Okay?
I'm not sure I understood this. So the source of the stochasticity is by discretizing the diffusion into smaller boxes,
No,
having molecules jumping back.
that's only part of it. The origin of the stochasticity is that there are few molecules in the system. Okay, so there are few sites at which you can initiate these new filaments. Okay, now your question raises a very interesting auxiliary question, and that is, G-actin is here in quite large numbers. How do I develop an algorithm that basically can treat some species deterministically and other species stochastically? And some people are working on this, okay, but it's still a large issue. Yes?
Okay, so the stochasticity comes from the nucleation of the filament. It's just there.
Yeah.
Nothing else.
A small number of those, right, is what gives rise to that. Okay. Finally, the exterior problem. Remember, the exterior problem was, can I move by shape changes, forgetting for the moment what the cell has to do internally to generate those shape changes? Okay, so we've had a little bit of an introduction to what the cell might have to do. Okay, the exterior problem. This is governed by what are called the Navier-Stokes equations. This is an equation in a deterministic description of a fluid for the velocity at a point in the fluid. Okay? And it has this derivative in boxes, which is similar to the Lagrangian derivative that Anatita showed earlier. And it's multiplied by something called RE, which is the Reynolds number. And the Reynolds number is extremely important here because it tells you what regime of the fluid mechanics you're really operating within. So for a large whale swimming at about 10 meters per second, the Reynolds number is this, 3 times 10 to the 8. At the other extreme, a bacterium swimming at about 1/100 of a millimeter per second, the Reynolds number is 10 to the minus, I think that's 5, whatever, it's small. Okay, what does that mean for that equation? Okay, firstly, it means that the balance of terms in this equation is very different for a whale than it is for a bacterium. Okay, and without implying anything, let me say that Onatita is more toward the whale than the bacterium when he swims. Anyway, what do you think the simplification would imply if I set that Reynolds number equal to 0? Well, for the mathematicians, everybody probably understands that this is what's called a singular perturbation problem. It's not strictly that, but it's like that, okay? Where some things vary rapidly and other things vary slowly. In this context, it's basically throwing out all inertial effects and saying that momentum diffuses throughout the medium instantaneously, okay? That's the implication of setting the Reynolds number equal to 0. That leads to what's called the Stokes problem for low Reynolds number flow, which is to solve this equation, which involves a balance of this term, which is the viscosity-dominated term, and a pressure gradient. And you want to solve this for an incompressible medium, so that's what this condition means. And you want to do that for a specified sequence of shape changes at the boundary. So what we want to do is take this object, which we'll call a cell. We want to specify its velocity on the boundary and then ask under what conditions can it swim. Okay? And in particular, when can it propel itself by shape deformations? It's easier to say when it can't. Okay? This is a nice theorem due to Ed Purcell from the 70s called the scallop theorem, which says if the sequence of shapes in a cyclic time periodic stroke is identical when viewed under time reversible, then there's no net motion. Because, you see, time plays no role in this equation. Time has been thrown away by setting Reynolds number equal to 0. Okay? So, if the only motion that a scallop could do would be to open and close its shell like this, it couldn't move because it only has 1 degree of freedom, and however you view this, this is reversible. Okay? Fortunately, scallops don't do mathematics. Turns out that starfish prey on scallops, and they move very effectively. Okay? So what do they do differently? Well, when the shell is closed, it pumps fluid out of the rear through a sequence of jets. So it's always moving in the direction in which the thing is opening and closing. Okay? So how does one look at this mathematically? Okay? Okay, I'm almost out of time. So let me go through this very quickly. If you're interested, there's a paper that will be coming out in Journal of Math Biology with Qixuan Wang, my former student. But the main idea is relatively easy to understand. You have to solve these Stokes equations subject to the constraint that there is no net force and no net torque on the object. Okay? Now you can worry about why no net force, no net torque at your leisure. Suffice it to say that it comes out of this assumption of setting Reynolds number equal to 0 and diffusion of momentum being instantaneous. In any case, in 2D, you can do something very simple. That is, you can reduce the Stokes equation to a single equation called the biharmonic equation, whose solution has been known since the time of Gaursat. Okay? It can be represented in terms of 2 complex functions, all right, called the Gaursat function. So to solve the exterior problem in 2D is simply a matter of finding those 2 complex functions. But simply a matter is not simple necessarily. Okay? So what do we have to do? Okay? Well, first of all, once you know those functions, all the physical quantities that you're interested in can be recovered. This is actually somewhat similar to what Onatita was saying. See, this object is changing its shape. Think of what I showed you for Dictee undergoing enormous shape deformations. And now you want to solve for the fluid dynamics in the region exterior to those shapes. That's very complicated, okay? So what you do is map it back to a fixed domain, solve on that fixed domain, and then map it forward to get the actual motion, okay? And 4 or 5 of these slides are involved with doing just that, and I'm out of time, so I will go through this very quickly, but I'll certainly be happy to tell anyone the details if they're interested. Let me just jump to some of the results. What we're interested in is to try to see whether dicty really can swim in the way that Peter von Hastert suggests, namely by putting out a protrusion at one end, propagating it around the boundary, and then reabsorbing it. Okay? That was the von Hastert picture. Put out a bump, as he calls it here, propagate it around, and absorb it down there. And the question is, can that be done? Now, a first difficulty is that it's pretty hard to generate those kind of shapes with some of the standard functions in complex variables, Laurent series, for example. So we do a Schwarz-Christoffel transformation. You forget the details. Okay, what we're going to do is we're going to compare 2 different cell types. Peter is the one that identified the 3 different modes. Barry was the person whose video I showed you of the swimming. The cell type is the same, but of a slightly different, what would it be? Genre? I'm not sure what the technical term is. So they differ slightly in aspect ratio, etc., the period, etc. Okay? So you can look at how a variety of different factors affect the motion. Oh, let me go back one. I think I want to show you, whoops, a computational result here. So here's just one example of the fact that they can swim, and this is makes a distinction between the number of terms we take in the Schwarz-Christoffel transformation, and you see the smoother it is, the faster it goes. But the main point is that Dicty figured this out long ago, but it does work by propagating.
Does it need the 2 protrusions, or
Yeah.
is enough with 1? Can it swim with 1?
Well, if I had time, I would. Let me just tell you what the result is. If I only have 1 and I oscillate it across the side, then it goes forward like this instead of smoothly, okay? But it still moves, okay? And I have a video of a comparison of the different modes, but I won't have time to show that. Okay, so you can look at all the different factors, the aspect ratio, et cetera, and see how it affects the motion. And the point here is that one can begin to say something that may be of interest to the experimentalist in terms of how do you understand what a cell should do in order to move through a certain type of medium. And to finish off, let me show you an example in which there are actually obstacles. This requires a different computational technique, but I won't go into it. So we have the no obstacle in the center, a fixed obstacle at the top, and a movable obstacle at the bottom. So the movable obstacle is simply floating in the fluid, and the hydrodynamic interaction between the swimmer and that obstacle moves it. And so you see, if the obstacles are free, he doesn't suffer a huge penalty. Trying to get through fixed obstacles is a bit more difficult. So what do you think might be the relevance of this to understanding movement of cells in the ECM? Well, it tells you, for example, that if the cell is moving in a confined region that is more or less fixed, it's going to be a great deal more difficult to move than if it's in free fluid. But if the extracellular matrix or whatever the environment is can, in a sense, breathe as the cell gets through, then, of course, the cell can move more readily. Okay? So, I guess time is up. So let me just summarize by saying what I've tried to do is indicate what some of the processes are that are involved in a more integrated description of cell motility. We're going to hear a lot of details about various of these throughout the next 2 weeks. So I hope this will kind of set the stage for you to begin thinking about perhaps questions that you should be asking in later talks. So with that in mind, let me just acknowledge the people that do the work, as usual. Whoop. And those are Yogan Cheng, who is working on the direction sensing. He's a postdoc. Xishuan Wang, who worked on continues to work on the swimming cells. And by the way, there's an interesting result that has come out of recent work. I told you that the scallop, which has only 1 degree of motion, can only move, or it cannot move if that was the only thing it could do. So you might ask yourself, if a cell can only change its volume by increasing or decreasing it, could it move? And the answer is no. There's only 1 degree of freedom. But if you put more than one cell together, through the hydrodynamic interactions, they can swim. Okay? So there's a result which still needs a lot of work. But anyway, thank you for your attention.
I'll take questions. We might have 5-minute break, and then we have a discussion. And at the moment, there's no set framework of what type of discussion we have, but we can start now.
So Hans, the original piece of work you showed where your cell gets excited all the way around and then there's symmetry breaking in the front, senses more than the back. A few questions on that. Is there a biological reason why that's beneficial? And secondly, having done the maths, can you shed a bit of insight into the counterintuitivity? Or is it very technical?
So what was the question? Why it extends to?
So yes, so what? Because you have this uniform sensing around it.
Yeah.
Is there a biological reason why it does sense all around it?
Oh, you mean in the direction sensing, why the initial response is spatially? That's a very good question, and my only interpretation of that is that the first response sort of primes the system, and then it's the secondary response which really establishes the direction. And beyond that, I can't say. It's interesting that you have to have the right kind of localization of species within the secondary response to produce that symmetry breaking, and that's induced in part by the initial uniform response. So that's probably the best I can do in terms of why that would be useful. Yes.
The follow-up question. So when you showed comparison experiment and your model, in experiment the first response was identical for the front and the back, but for your model the one was higher to begin with, and that makes me suspect that you don't have spontaneous breaking.
Well, very good question. Experimentally, what they display are not pointwise values around the cell, but an average of the front half versus the back half. Okay? So what they're doing experimentally is they're averaging over the front half and then averaging over the back half, and that's what front and back means. Okay? But we solve it pointwise in the whole domain. So that could account for part of the difference. What I thought you were going to point out was that the time scales are slightly wrong. Our system is a little bit too fast, okay? But mathematically, that's easy to deal with. But the more interesting question is, where in the biology should changes be made, or in the description of the biology should changes be made to account for that time difference? Okay, and that comes to an issue that comes up again and again and again in complex models of biology, and that is very rarely do you have all the parameters you need in a complex model. All right, so someone can ask, what good is it? Well, what you can do as a mathematician is to identify the sensitivity of the response to various parameters. All right, and then for one thing, you can say to an experimentalist, here's where you should focus. If you believe my model, here's where you should focus your effort to get better parameter estimates, because these are the ones that are really controlling the response, and others are kind of along for the ride, so to speak. Okay, so that's a sensitivity kind of analysis that one should do. Yeah.
My question comes exactly to this point. Your model is already a simplified model because there's no way we can model this maybe 300-400 species of molecules interacting to form, for example, these actin waves. Now, as a physicist, I'm trained to try to measure all concentrations that come into your equations. In a perfect world, at some point we would be able to do that. But now comes the fundamental problem that in your model you have something like effective concentrations because you have lumped different molecules into one species effectively. Is there a rational way to map this into the huge complexity we are facing?
Well, one way to approach it might be effectively in the other direction, and that is to start with a more accurate, more detailed model, and then try via identification of different timescales, etc., to identify a reduced model which captures the essential behavior. Okay, and that's a point at which I think sometimes a cartoon model can be quite useful, in that you put together a cartoon model that has a certain number of processes in it, you can understand what dynamics it predicts, etc. You can moreover frequently understand what it is that controls the different aspects of the response, right? So in Fitsunagumo, you know, for those of you who know the equation, you know, the cubic part looks like u, a minus u, u minus 1, etc. If a is very small, it's very excitable. If a is large, it's very inexcitable, etc. The point is that it takes a whole, what shall we say? Well, Hillary Clinton once said, it takes a village to raise a child. Here, I think it takes a village of techniques and models to eventually really describe a phenomena. And there is no algorithm that says if you do this, this, and this, you will arrive at the correct answer. But there's a lot of intuition and experience and so forth that goes into it. Yes?
Yes. Actually, I have a more biological question. You describe the waves of actin assembly, which are quite irregular, at least as can be seen here. At the same time, for cells, for some types of cells, are typical very regular wave. For example, if you know what podosomes are, there are contacts of some types of cell with the matrix, quite uniform size actin kind of aggregates, which however behave as waves, and some of them disappeared, others are formed, and this in a way, the question is, to describe these more regular systems, you need kind of more complex assumption, or maybe this kind of type of organization can be derived from also some simple assumption, and you will obtain this numerous actin polymerization kind of devices of uniform size, which are, however, behave as waves.
Well, here's one possible answer. In the experiments that I showed, and Till can give you much more detail, you first knock down the actin network. So you're basically looking at the reconstruction of the network, and that will inevitably have a lot of variation in it, spatially and temporally. Whereas in the system you're describing, you're inducing these new structures in an already very structured environment. And that may account for the higher regularity.
I do not think so, because you just have cells that have no contacts whatsoever. You plate it on the uniform substrate, and then you obtain this periodic
Yeah, but the cells in that context have a well-developed cortex, and the actin, the actin cytoskeleton is there, right? It's true that your contacts with the surface are, a few, right? But I don't know if that's the answer. I mean, it'd be an interesting computational problem to try as to whether or not you can see that regularity in that situation. I mean, are these focal adhesions at the cells?
No, I'm speaking not about focal adhesions. I'm speaking about much kind of simpler adhesions called podosomes, which normally are of uniform size, but in some cases they could be much larger, and in this case they called invadopodia and typical for cancer cells.
But what's the interaction with the substrate? Is it van der Waals?
The substrate exactly like focal adhesions.
So it's integrins,
by
etc.
integrins
Okay.
and so on. The difference is that focal adhesions require force to assemble, while podosomes not, because they can actually be formed on a fluid substrate.
Okay.
So
I mean,
it's kind of
focal adhesions only require the force to actually build up. I mean, that's the positive feedback loop.
Exactly. This is something else.
Okay, it's an interesting question, and that's exactly what we're looking for. Okay, so some theoretician should take note of that and ask, what can I do? Okay.
Any further questions? One more? Yeah.
So I had just one question regarding the swimming of cells, the development and movement of the cell, of the protrusion on the cell surface. So is it within the Scalp theorem? Is it time reversible, or it was not? I was not sure whether this is the case.
No, it's not. And besides which, there are always, so I showed you the symmetric case where there are two, right? So there are, in a very simplistic way, there are at least 2 degrees of freedom in that system, because each of these can move, okay? So it's not...
So can it swim even within the Scalp theorem, like if you set up some time reversible development of a protrusion?
Well, maybe, yeah, I think I see what you're missing. There are many, many degrees of freedom in this shape, right? Because we have to mathematically describe the shape and the velocity at all points along the surface. Of course, we discretize, but we have some finite number of points, so there are many degrees of freedom in the context of the Scalp theorem. I mean, it looks like if you only have one pseudopod, there's only 1 degree of freedom, but there's a lot in the background.
Let's thank Hans again.